Hello and welcome, everyone. Thank you for joining us today. I am Dr. Carlos Vasquez. I'm the CEO at FASI, and it's an honor to welcome you to another great discussion on our seminar series. At FASI, our focus is to understand, translate, and intervene with food allergies. And one of the key questions that our research is looking to answer is why is there such a big increase in food allergies over the past few decades? In September, we introduced the seminar series that we call Project Safe, the impact of chemicals in our food and the environment. And this series highlights some of the evolution of the research that we've been doing in terms of understanding how chemicals in food and the environment could have an impact on human health, and specifically how that rapid increase in food allergies could be associated, and other chronic disorders could be associated with some of the chemicals we experience through the environment and the food that we eat. And specifically, one of the key areas is to understand how, excuse me, how some of the chemicals in the food that we eat could be acting as adjuvants or agents that can promote and activate the immune system. Well, today I am really excited to have with us Dr. Wolfgang Lettner, who's the Chief Innate Immunity Section lead at NIH. And Dr. Leitner has been working in this area of understanding and characterizing adjuvants for the development of therapies for more than a decade. And so when we think about this problem, we've been talking about adjuvants in the context of food. We typically, and we haven't actually been talking yet about the development and the characterization of actual adjuvants in the context of rational design of therapies. So it is an honor to have Dr. Leitner with us, who has been really looking at this question in a very focused way for way over a decade and brings so much knowledge to the table that we hope to help us understand what are adjuvants and how do they work, and now how could that be associated with food that we're eating nowadays. So with that, Wolfgang, it's a pleasure to have you here. Thank you for joining us and really looking forward to having a great discussion.
Thank you so much for the invitation to talk about adjuvants. So our adjuvant at NIAID is designed to establish a robust pipeline of new novel immunostimulators, help them get into the clinic, but also establish how and why they work. We recognize that there are very few adjuvants that are in licensed vaccines. And so we're trying to help vaccine developers to gain access to novel adjuvants. And it's not just access to adjuvants that are that can be taken into the clinic and then into product development, but also the right ones, meaning the ones that induce the most appropriate, meaning the most effective immune response against a particular pathogen or disease that's being targeted. And to help with that effort, we've actually created an online database. It's called the Vaccine Adjuvant Compendium that introduces vaccine developers to accessible adjuvants. So please check it out when you get a chance. My talk today, however, is going to be very different from what I usually talk about because our program is primarily focused on infectious diseases, and I am not a food allergy expert, just to get that out of the way. So let's get started with talking about the need for and the importance of adjuvants in vaccines. So when you look at what's in a vaccine, the four primary ingredients, the target antigen that induces the antibodies and the T cells to the pathogen, an adjuvant that induces the innate immune response, which allows those T and B cells to be activated. And that adjuvant is either present naturally or is added to the vaccine exogenously. And then preservatives and stabilizers that protect the ingredients from both from contamination and degradation. So even though from an immunological standpoint, both antigen and adjuvant are active ingredients, the regulatory agencies only consider the antigen of the vaccine to be an active ingredient. That's mainly for historic reasons. So when you look at any of these descriptions of or depictions of vaccines like this one, the focus is usually on vaccines for pathogens, but vaccines are also being developed for a much wider array of conditions that includes cancer, allergy to pollen or food components, vaccines against illicit drugs, for example, opioid vaccines, or to prevent transplant rejection. And so that means the expectations for what an adjuvant should do exactly in a particular vaccine will differ dramatically between these different applications. So when does a vaccine need an adjuvant? When you look at a vaccine against pathogens, a really simplistic way of answering that question is the dirtier a vaccine, the more immunogenic it is, and therefore you don't need an extra adjuvant. And by dirty, I don't mean dirty in the classic sense, but how many of the molecules that are associated with the pathogen are still in the vaccine. So for example, as listed here, LPS, the lipopolysaccharide, or nucleic acid, bacterial DNA, viral RNA. And those have adjuvant activity. And also, how is the vaccine seen by the immune system? Is it seen as something benign or as a threat? So if it replicates, meaning if it is an attenuated pathogen, that activity, that replication by itself also provides an adjuvant effect. So the cleaner a vaccine, meaning especially those recombinant proteins, the less immunogenic they are, and therefore the more they depend on the presence of an exogenous adjuvant that you add to the vaccine formulation. But that's not to say that only those need adjuvants. Even those traditional vaccines can still benefit from exogenous adjuvants. This is a topic we could talk about for hours, but the only example I want to mention here, because it came out of NIAID's adjuvant program, is the COVID vaccine Covaxin. This is an inactivated virus that was adjuvanted with L-hydroxyquem 2. This was the first ever TLR7/8 adjuvant that was used in an infectious disease vaccine. Was it necessary? Would the vaccine not have worked? It would have worked, but by far not as well as with the adjuvant. So let's take a step back and ask, what is actually the definition of an adjuvant? Both from a scientific and a regulatory perspective, they're not defined by what they look like. They're not defined by what they're made of. They're not even defined by how they work, so their mechanism of action, but by the outcome of the response that they induce. So in other words, the adjuvant effect. And that adjuvant effect is their ability to enhance T cell and B cell responses, so making good antibodies and good effective T cells. So historically, adjuvants come from natural sources, primarily pathogens and derivatives of those molecules, but also from some unexpected sources, such as plants. And that's going to get us into the topic of adjuvant-like molecules in food. The bottom line is that anything that triggers an innate immune response could be a vaccine adjuvant. So these could be actual physical molecules that bind to receptors of the innate immune system. Anything that kills host cells and therefore causes the release of molecules from these cells that now are recognized as being out of place and therefore associated with some kind of a danger. Any mechanical or chemical stimulation of tissue. So think of tattoo guns or the gene gun that's used to deliver DNA. Those are mechanical insults to the tissue that provide a really good adjuvant effect. But even energy, so for example, electrons or photons could be acting as an adjuvant, as in the case of the adjuvant laser. So all of those signals, all of these molecules trigger downstream signaling events. And those mediate a wide variety of innate immune signals and changes. And in the end, they all lead to the promotion of T and B cell responses, so the actual adjuvant effect. So to get to the point of this presentation, where is the link between what's used in a vaccine to enhance the response of the vaccine and food ingredients? Or more precisely, how and why is there a link? What you're seeing here is probably not a comprehensive list of adjuvants that you also find in food, but they're probably the most common ones. So let's start with the oldest adjuvant in human vaccines, and those are aluminum salts, so collectively known as alum. They're also the most common adjuvants in human vaccines to date. If you think about the amounts, it's often brought up as a concern. When a child gets multiple vaccines that are all adjuvanted with alum, the actual amount of aluminum per shot is less than 1 milligram. Now, you find aluminum and aluminum salts frequently in food. They're either there naturally because plants pick up the element from the soil, or because they're added. Think about food coloring. The recommended maximum safe dose is, again, 1 milligram, but per week, per kilogram of body weight. So what we're injecting is much, much lower amounts. Then the next one is, so this is in the category of metals. The next one is in the category of oils. Squalene is a ubiquitous oil. There's lots of it on our skin. The main user is the cosmetic industry. By itself, squalene is immunologically inert. So the immune system does not see it. So technically, by itself, it is not an adjuvant. It has to be formulated with other, also immunologically inert substances to create an actual adjuvant. And I'll get back to that point in a moment. There's a number of foods that contain, some cases, even large quantities of squalene oil. The next category is glycosides. Glycosides are plant-derived molecules, also called soap molecules. Most famous one comes from the Chilean soap tree. Saponins are used, specifically one of them, is used in both veterinary vaccines and human vaccines. In case of the human vaccines, the examples are the shingles vaccine. One is they're also in one of the RSV vaccines and one of the COVID vaccines that are licensed in the US. They are abundant in some food. So think about when you rinse quinoa underwater, it creates a lot of foam. That's saponins. They're added to foods. Think about the foam on Coca-Cola, especially the foam on root beer. It's all coming from the same source, the tree bark of the Chilean soap tree. Now, what might be really surprising is that sugars can be great adjuvants. And I want to mention two families of sugars, starting with inulin. Now, inulin in its adjuvant form is a crystal. And in food, it is a soluble molecule, whereas the crystal is insoluble. And it is in food, it is a ubiquitous food additive. I'll get back to that in a second. The second huge family of sugars that can be used as adjuvants are glycolipids. It's basically a sugar with a lipid tail. They are kind of a new frontier for adjuvant research. And why is that? It's mostly because the receptors for those types of molecules have only been discovered relatively recently. So they're ubiquitous in food, used as emulsifiers, as stabilizers, and so on. So this is where the overlap is, as far as I can see it, between the vaccine world and food. Now, so the question is, how can a sugar be an adjuvant? And to briefly address this somewhat paradoxical observation, we have to look at the structure of the molecule. So inulin is sold in bulk at health food stores. It's added to yogurt. The big So inulin by itself is a polymer of different sugars. You can see this here on the side. It's a global commodity. It's also used medically. It's used to test renal function. So you inject it IV and then you measure how much of it is cleared in a certain period of time. Now, in its natural form, in its soluble form, that sugar is completely immunologically inert, does not stimulate the immune system. However, if you create a crystal, a water-insoluble crystal, it now activates the immune system, has been shown to be a great partner for adjuvants. And one of those adjuvant combinations of this sugar and a piece of synthetic DNA is used in yet another COVID vaccine that came out of our program. It's called Spikogen or COVAX-19. And that one's used to not only protect humans from COVID, but also zoo animals. So what kinds of receptors are there that recognize sugars as something that should trigger an immune response? And turns out that we express a pretty wide variety of receptors that bind sugars. So collectively, they're called lectins. A subfamily of them are the C-type lectin receptors. They include, those are shown here, a variety of innate immune receptors. And those have evolved to specifically recognize the sugars that are typically associated with pathogens. And they are among the newest targets of adjuvant research. So what about those two sugars that I just mentioned? Turns out that the crystalline inulin binds to this receptor right here, DC sign, and glycolipids bind to another subclass of those lectins called lectin-1. And shown here are just two of the countless glycolipids that are out there in nature. And we still don't know enough about them yet, which one binds to which receptor and how all of these receptors work and are different. So now that we've seen the overlap between molecules used to stimulate vaccine responses and components of foods, let's talk about the elephant in the room. And that is, why wouldn't a molecule that is great at stimulating an inflammatory response when you inject it in small quantities, why wouldn't that also not cause an inflammatory response in the gut when you ingest much, much larger quantities, and vice versa? If you put food ingredients in vaccines, could that lead to food allergy? Now, the short answer is, obviously, that's not happening because food will inevitably contain, to some degree, certain microorganisms. And those organisms could be either undesirable bacterial contaminations, but also think about like fungi in blue cheese. They will contain lots of immunostimulators. In addition, the human body contains more bacteria than mammalian cells. Most of them are part of the microbiome in our gut. And those are microorganisms that, again, produce lots of immunostimulators. So how could molecules that allow to protect us from a deadly disease by stimulating very strong immune responses, how can they be safe when you eat them? There are probably more mechanisms involved in that than I'm showing here, but here are the two big ones. First of all, location. Where is the immunostimulator encountered? We expect to see a lot of these molecules in our digestive system, and so there's no point in constantly responding to them. So the strategies to avoid inflammation would include, number one, not expressing the receptor that would see them, or if those receptors are there, you generate a completely different signal than when the receptor is engaged when in the periphery, meaning it's a completely different response to something that you see in your gut and seeing the exact same molecule in your bloodstream. However, when the intestinal integrity is compromised and now immunostimulatory molecules or entire bacteria from the microbiome are able to penetrate that barrier, they can indeed trigger an undesirable inflammatory response. Now, in addition, those immunostimulators in our food are likely affected by the harsh conditions in the stomach first, and then by the digestive enzymes in the gut. Now, the second important aspect that I already mentioned is formulation. So formulation means how the immunostimulator is presented to the immune cells and how it is seen by the immune cells. So for example, some innate immune receptors need to be cross-linked. That means you need two molecules, two agonists of these receptors that are somehow linked and now bring two copies of the receptor together, which means if the single molecule binds to the receptor, nothing happens. Some adjuvants may consist of components by themselves or weakly or not even immunogenic at all. And I mentioned squalene before as a classic example. So what I'm showing you here are a couple examples of adjuvant formulations. The first one is liposomes. Those kinds of delivery vehicles are used in the shingles vaccine. And what you're seeing here is liposomes carrying either in their lipid bilayer here, or if they are water soluble, carrying the adjuvant and antigen in their core. Another approach is using these cage-like particles. Those are the adjuvants in the COVID vaccine from Novavax, or oil and water emulsions. The oil in this case being squalene. That kind of adjuvant is used for the Fluad influenza vaccine. Another approach is to use polymeric biodegradable particles that by themselves may not be adjuvants, but they can deliver both antigen and adjuvants and release them over an extended period of time to improve the immune response. And the other approach I mentioned before for the receptor cross linking is to multimerize small molecules. So in yellow, you see the actual adjuvant, and in blue are linkers that bring multiple copies of the adjuvant together to actually make them immunogenic. So the last aspect I'd like to address is the use of adjuvants for vaccines against food allergy. Most of the adjuvants, and that includes most of the adjuvants from our program, are at least initially designed to enhance immune responses, to potently stimulate innate immune cells, namely the antigen-presenting cells, APCs, drive up antibody titers, and induce very strong T-cell responses. And those are the kinds of adjuvants you would use for example, infectious disease vaccines or substance abuse vaccines, where you want lots and lots of antibodies neutralizing the target antigen. Now, that's why the error is the biggest. That's the main focus of adjuvant research. Now, at the beginning, I mentioned that adjuvants should be selected not only based on how potently they enhance an immune response, but also based on the profile of the immune response they induce. And we can use adjuvants to drive vaccine-induced immunity towards a desired immune profile. So, for example, in case of allergy, you may want to redirect from the allergic Th2 response to a non-allergic Th1 response. For autoimmunity, you may want to direct the response away from the tissue-destructive Th1 response. For a tissue-destructive Th1 CD8 T-cell response. Now, similarly, we may be able to drive food allergy with specific adjuvants. And right now, we're actually supporting a study to select the most appropriate adjuvant for a peanut allergy vaccine by systematically comparing different adjuvants side by side to see which one induces that desirable immune profile. Now, the most challenging approach, however, and that's why it's still at an early developmental stage, and this is why the arrow is skinny, is to use tolerogenic adjuvants. This would be for conditions such as food allergy or, for example, to prevent transplant rejection. So these types of adjuvants are kind of paradoxical because they don't induce an immune response. Instead, they actively suppress the response in an antigen-dependent manner, hopefully for long periods of time. So this is very different than using, let's say, corticosteroids to suppress immune responses. There are different immune mechanisms that our immune system uses to prevent adaptive immune systems, so B cells and T cells, from responding. And those are summarized here. The desired outcome is that an antigen-specific T cell that could recognize, in this case, the allergen, the food allergen, to become either paralyzed, killed, or being actively suppressed by a subtype of T cells, the T regulatory cells, that prevents the activation of other T cells that recognize the same antigen. We don't know yet how adjuvants would work and which kind of outcome they would produce. Doesn't really matter as long as they are preventing the induction of the allergic response. So to finish up, I won't go through the details of this slide, but I just wanted to show them to illustrate the challenge. So on paper, in theory, we know how to make a vaccine for food allergy using tolerogenic adjuvants. However, as you probably all aware, there are no tolerogenic adjuvants in any kind of advanced vaccine development program, simply because we're still stuck at the earliest stage of that process. We do know how to activate, meaning how to turn on an antigen-presenting cell with an adjuvant so that it is subsequently able to induce good T and B cell responses. However, what we don't really know is what an APC, what one of those antigen-presenting cells would need to look like that is capable of actively turning off those T and B cell responses. So in other words, a tolerogenic APC. If you're screening for tolerogenic adjuvants, if you're trying to discover them, you do need to know what to look for. You need to know what are the features of one of those tolerogenic dendritic cells, so you know which compounds, which types of tolerogenic adjuvants to go after. So here are the features, the summary of the features that we need to go after. And on the left side, the phenotype of a tolerogenic APC. So it has to be resistant to maturation, meaning it can't just snap out of that tolerogenic state and then activate T cells and make matters worse. It should express inhibitory receptors when it's interacting with a T cell, but not the receptors that are associated with stimulating T cells, so meaning co-stimulatory molecules. Instead of cytokines that are stimulating T and B cells, these tolerogenic APCs need to produce completely different immunomodulators, such as interleukin-10 or TGF-beta, which are known to be immunosuppressive. One of our adjuvant contractors, it's the biotech company Corona, they're currently working on identifying the minimal set of robust characteristic markers of tolerogenic adjuvants. So picking from this long list that you're seeing on the left, picking those that can be used in a high throughput screening campaign to screen for inducers of that phenotype and then develop them into adjuvants for a vaccine against celiac disease. With that, I'd like to end the presentation. Also refer you to the website of the NIAID adjuvant program. It describes the various ongoing efforts to discover and further develop new vaccine adjuvants. And I'd be happy to answer any questions you have. Oops.
Thank you so much, Wolfgang. This is really exciting, and it's great to have someone like you from the NIH that have been really focusing on understanding adjuvants for such a long time to tell us more about those details. Before I jump into questions, I just want to remind people, you can people ask us, how can you help? There's a QR code here. Follow us in social media, donate, host an event, and join our webinars. And our next webinar would be actually with one of our scientific advisory board members from Stanford University, Beth Sadley, and that'll be on May 6th at 12 p.m. So I hope people can join. Briefly, Wolfgang, I was really impressed by that table that you showed in comparison of how adjuvants are found, are typically naturally occurring. Some of them, just stimuli, right, on the skin and lasers. and how some of them have made their way into actual adjuvants in vaccines. But what I gather from it is the devil's on the details, right? We've been talking in this seminar series about molecules in food that could act as adjuvants. But what I guess probably one of the things that I highly appreciated from your conversation is that it's not about only the molecules found in something, but how the quantity, the timing, the configuration. Some of them may be fun, but if it's not in a crystal structure, they're not going to elicit the adjuvant effect. So it is actually very helpful to see this for for our audience that have been listening to molecules that can act as adjuvants can also be in food. There seem to be a lot of things that need to come together to elicit, even when you're trying to design them, right? To use them in vaccines, unless you put them in the right way, they're not going to have the effect that of adjuvant effect that we're talking about.
No, so in theory, you could take a vaccine vial and eat it, and nothing would happen, right? Because you're putting it in the wrong place. Even though that they contain, formulated adjuvant, the quantities are wrong. The vaccine's going to be digested in the stomach. Nothing immunostimulatory is actually going to end up in the gut. With vaccines, we know that depending on where you deliver them, you will get different responses. Whether you inject them in the muscle, in the skin, um, if you inhale them, you use them as an intranasal vaccine, you get very, very different immune responses. So coming back to that point, location, 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 where you encounter an immunostimulator is extremely important. That's not to say that there can be, pro-inflammatory components in food. But want, what I want to highlight is just because the names sound similar doesn't mean that it will have the same effect. And I think inulin is like the prime example. Chemically, it is exactly the same molecule, but depending on how you prepare it, whether you turn it into a crystal or you leave it in its soluble form, it's a difference like day and night. One is an adjuvant, the other one is a completely immunologically silent molecule it's food. It's a sugar. It's being digested. It's turned into energy. It has absolutely no impact on the immune status, even of an immune cell. So you could feed it to an immune cell, and it would not change its immune status. So I think that's the really the key point is context matters. Like you said, quantities, location, how you show the molecule to the immune system and where.
No, this is really good that you put it in context, Wolfgang, because I know many of our audience, right? It now, for example, you talk about squalene, right? Which I know it's in many personal care products, and that you put on your skin. And to your point, it doesn't mean that by having the actual molecule there could lead to an adjuvant effect. Yet at the same time, there's, also new research evolving on maybe there's some particular conditions that may, as you said, may trigger an inflammatory response. And there's been some publications coming up from our investigators recently, right, that says, hey, if you have an inflammatory response here, it may accentuate the response to an allergen when you're eating it as well. So a lot of research to be done, but I guess what I gathered from this is the devil's on the details, right? It's not just having a molecule, it's the presentation. So that's why it's so important for all of us to continue to explore the biology and the relationship, the structure activity relationship between these molecules and the biology, especially the biology that can trigger this. Coming back to the question that we've been trying to answer, why is there such an increase in food allergies over the many years? And are there some new biologies that are under explore, or some new chemistries that are under explore that may somehow come together to synergize and provide these responses? But from someone that has been doing this for a long time, and you're really a master on this and understanding what it takes, it's difficult. It's a very difficult natural process in order to do this and kudos to you and your team that have been working on this for a long time. I unfortunately, I know we don't have, we're out of time. And there was only one, I'm going to try one quick question from Jimena here. What are alternatives or procedures that are available for children allergic to vaccine ingredients? So I guess many people are allergic to vaccine ingredients. Sometimes is it the adjuvants, and what are some of the alternatives?
So there aren't allergies to the adjuvants that I'm aware of. The allergies tend to be to ingredients that are not really essential for the vaccine. So the classic one, of course, is egg allergies. And that is a problem with vaccines that are being made in eggs. The most prominent example being the flu vaccine. The alternative in that case is switching to a vaccine that is not made in eggs. So in case of flu, you would switch to the recombinant protein. That vaccine is called flu block. It is made in it's made in cell lines. So for some vaccines, unfortunately, you don't have that luxury where there are multiple versions of a vaccine against the same disease. The luxury of just being able to switch to a completely different vaccine. Um, as novel vaccines are being developed, you will see fewer and fewer being made in a context that creates this kind of problem. In other words, there's a push to get away from egg manufacturing of vaccines for multiple reasons. It's much more cumbersome. It's easier, more straightforward to make them in cell lines. Unfortunately, right now that the only way around that is you have to avoid those vaccines because they will, of course, create a safety issue for the children who are allergic. But
Great.
step one is see if there's an alternative vaccine that's made differently that does not contain the allergen.
Good. That's great. Thank you, Wolfgang. And thank you, Jimena, for your question. Well, I know we're out of time, so unfortunately I cannot take any more questions today. But Wolfgang, a real pleasure to have you. Great work, and thanks for teaching us about adjuvants today. Definitely a great piece to incorporate into our other discussions around this topic.
Thank you so much for having me.
And thanks to everyone for joining today.